So I'll describe the process by which we developed the study. And the methods here were pretty rigorous. We started with a literature review that looked at both the preference elicitation method and the identification of the treatment attributes. And then key to the entire process was the advisory board that Isabel just described. One of, I think, the biggest strengths of this study was having such a diverse advisory board throughout the process that helped us ensure that we were staying on track, answering the questions that mattered, and including the features that mattered not only to the people with hemophilia, but to the decision makers as well. Prior to initiating the survey, as part of survey development, we did 20 interviews in Belgium with people with hemophilia to look at what information needs they had and their understanding of gene therapy, the factors that would influence their choice of treatment for hemophilia, including gene therapy, and then we identified the top treatment features for hemophilia, assembled these together, and then we administered the survey to 117 people with hemophilia in Belgium using the threshold technique. So as an overview of both of these phases, the first method included semi-structured interviews with both open questions and a ranking exercise, the objective being to identify the treatment features that mattered. There were 20 interviews among people with both hemophilia A and hemophilia B, most with severe disease, and this comprised 4.5% of the population of people with hemophilia in Belgium. And for the full survey, it was a fairly standard patient preference survey looking at including demographics, disease and treatment history, quality of life, and a preference elicitation technique. But one of the things that was unique about this study was it included an interactive educational tool, which I'll talk about in a moment. And the goal was to look at what the trade-offs were that people with hemophilia would be willing to make when considering a gene therapy. And the survey was 117 people, which is on the smaller side for patient preference studies as a whole. But it's important to note that this is a rare disease, and this comprised 26% of the population of people with hemophilia in Belgium. So I had mentioned before that one of the unique aspects of this study was the educational tool. And the educational tool was actually kind of fun to see. It was an interactive tool, but it was developed in a very deliberative process. The goal of this was really to answer a research question that IMI PREFER had put forward, which was, what is the impact of educational tools on outcomes in patient preference studies? So we did a literature search to understand not only the information needs that were important, but also how to structure the tool. We had interviews with patients. Again, remember we talked about information needs. That informed the tool. We asked hematologists to validate it for accuracy and to ensure that we weren't missing anything. We asked the patient representatives on the advisory board to help us make sure that it was correct, and then we piloted it. The tool itself was animated, as you can see here, and it was interactive. The animations were actually not static, they were dynamic. There was a voiceover to help people understand and not only have to read, and it covered 5 different modules so that people would understand how gene therapy works, what it generally does, how it is administered, what the effect might be, and what the side effects and uncertainty might be. So the next question that we had to answer was, for the full survey, which was the method that we would use? So again, this followed a fairly deliberative process. We started with 9 promising methods from the work that PREFER had done. We looked at that in relation to the research question, the relatively small patient population, the decision context, and the requirements that we were going to want to impose to establish the validity of our results. We then sought expert opinion on methods and came up with 2 methods that would be worthwhile or promising in this particular case. One was the threshold technique, the other was swing weighting. And in the end, we chose threshold technique for the fact that we can do it with fewer people, knowing that this was a rare disease. This was going to be an unsupervised survey, which made swing weighting somewhat problematic. And another thing that we'll see when we show you the questions is that this was a labeled design, meaning that we actually compared gene therapy to factor replacement as opposed to having an unlabeled design, which is common in most patient preference studies. Then we had to choose the attributes. So we started with a literature review. We had information and input from the advisory board. We tested those thoroughly in the 20 patient interviews and brought them back to the advisory board for clarification, because we had, as you will see, 5 top attributes, but there is confounding between factor level, bleeding rate, and the probability that prophylaxis could be stopped with gene therapy. So we had discussions with the advisory board about how to structure these 5, and in the end, it was determined that we would use 4 instead of 5. In other words, that we would not include factor level. So the survey looked like this. And the first question in a threshold question is what we could consider a direct elicitation, meaning that if these 2 options, prophylactic factor replacement therapy and gene therapy, are described in such a way that they represent a potential reality, this actually would tell us that given those options, which people would choose which therapy, and Aline will show you some of those results. So this is our starting point, and everybody started at the same place. Again, we talked that they were labeled. A is always prophylactic factor replacement therapy, which is standard of care, which is typically the left-hand side in a threshold technique. Gene therapy is the alternative. In this case, again, labeled as gene therapy. The primary differences in where we started was that the likely benefit would be a high probability of being able to stop prophylaxis. The risk of gene therapy in this case was that we had fewer years of information, so we had much more uncertainty about side effects. To be conservative, we assumed at baseline that there would be 6 bleeds per year in both factor replacement and gene therapy, and that the average quality of life reported in the clinical studies would be the same between the 2. So this was our starting point, and we were able to then say, given gene therapy with this particular profile, would you choose this over factor replacement? And if someone did choose gene therapy, in this case, it told us a few things. First, that gene therapy was an acceptable alternative. There will be some people who would not even consider gene therapy under any circumstances. It also told us that the probability of reducing the need for prophylaxis outweighed the uncertainty surrounding the side effects, given the fact that we had fewer years of time during which it was studied. And again, the bleeding and the quality of life were the same. Once we established the baseline, we then adjusted each of 3 of these one at a time to see where the threshold was at which someone would switch. So if someone chose gene therapy to start, we knew that we needed to make gene therapy look worse in order for someone to switch back to prophylactic factor replacement. If someone chose prophylactic factor replacement first, we would need to take gene therapy and make it look better to entice them to switch. So first we did bleeds per year. We then did the chance to stop prophylaxis, and then we looked at changes in quality of life that would be required, all as offsets to the uncertainty of having less information about the side effects that were studied. And with that, I will turn it over to Aline to describe the results.